Hello friend, my name is Ganesh Patharkar. I am student of Mo Education Society College of Pharmacy. Today I am going to try to explain the topic glycolysis under the guidance of Professor Sonone Madam. In biochemistry subject, we study the topic glycolysis in chapter carbohydrate metabolism. Glycolysis. We simply define glycolysis as breakdown of the glucose. But according to the first point, glycolysis is the catabolic pathway in the cytoplasm that is found in almost all organisms, irrespective of whether they live aerobically or anaerobically. The word aerobically or anaerobically argues very important information. That is, in the process of the glycolysis, the requirement of the oxygen is not compulsory to complete the process of the glycolysis. It means glycolysis was occurred not only in presence of oxygen but also absence of the oxygen. Second point, glycolysis is a simple metabolic process in which glucose is broken down in two molecules of the pyruvate, addition two molecules of ATP and two of NADH plus H plus are formed. Third point, energy gain in one complete cycle. In aerobic respiration, complete oxidation of one glucose molecule produce 38 ATP molecule, and in aerobic respiration, this is where ATP production stop. There is a final total of only 2 ATP produced per molecule of the glucose. Fourth point, glycolysis involve 10 individual steps, including 3 isomerization and 4 phosphate transfer. Only redox reaction takes place in a 6 step. From the next slide, we will going to understand the process of the glycolysis by step. First step of the glycolysis, phosphorylation of the glucose. Here in this step, glucose is converted into glucose 6-phosphate with the help of the enzyme hexokinase. It is the first priming reaction where the ATP is converted into ADP. Second step of the glycolysis, conversion of the glucose 6-phosphate. In this step, glucose 6-phosphate is reversibly converted into fructose 6-phosphate with the help of enzyme phosphohexose isomerase. It is the reversible reaction, and here we can also say that the glucose 6-phosphate is isomerized into fructose 6-phosphate. Third step of the glycolysis, phosphorylation of the fructose 6-phosphate to fructose 1,6-bisphosphate. In this step, the fructose 6-phosphate is converted into fructose 1,6-bisphosphate with the help of the enzyme phosphofructokinase. It is the second priming reaction where the ATP is converted into ADP. In this step, by using the ATP again, another phosphorylation takes place and it gives rise to fructose 1,6-bisphosphate. Here, enzyme phosphofructokinase first is the most important key enzyme in a glycolysis process. Fourth step of the glycolysis, cleavage of the fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is reversibly converted glyceraldehyde 3-phosphate with the help of the enzyme aldolase. In this reaction, cleavage of 6-carbon sugar phosphate to 3-carbon sugar phosphate is done. In this reaction, fructose 1,6-bisphosphate is broken down by aldolase into the C3 compound glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate. Fifth step of the glycolysis, interconversion of the triose phosphate. In this step, glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate is reversibly convert glyceraldehyde 3-phosphate second. With the help of the enzyme triose phosphate isomerase. Here we also find two phases, preparatory phase or payoff phase. In a preparatory phase where the glucose is phosphorated and cleaved to L2 molecule of the glyceraldehyde 3-phosphate, and in payoff phase oxidative conversion of glyceraldehyde 3-phosphate to pyruvate and the complete formation of the ATP and NADH. Sixth step of the glycolysis, oxidation of the glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. Here in this step, glyceraldehyde 3-phosphate second is reversibly converted into 1,3-bisphosphate glycerate. 1,3-bisphosphoglycerate second. It is the reversible reaction. In this reaction, glyceraldehyde 3-phosphate dehydrogenase is used as a enzyme. It is oxidation and phosphorylation in this step takes place. Here 2-pi molecule is also involved and 2NAD plus is converted into 2NADH plus H plus. Seventh step of the glycolysis, phosphoryl transfer of the 1,3-bisphosphoglycerate to ATP. Here 1,3-bisphosphoglycerate second is converted into 3-phosphoglycerate second with the help of the enzyme phosphoglycerate kinase. It is first ATP forming reaction where subtract level phosphorylation takes place. Here also the 2-ADP is converted into 2-ATP. Eighth step of the glycolysis, conversion of the 3-phosphoglycerate to 2-phosphoglycerate. In this step, 3-phosphoglycerate reversibly converted into 2-phosphoglycerate second with the help of the enzyme phosphoglycerate mutase. Ninth step of the glycolysis, dehydration of the 2-phosphoglycerate to phosphoenol pyruvate. In this step, 2-phosphoglycerate second is reversibly converted into phosphoenol pyruvate second with the help of the enzyme enolase. Here also the 2 molecule of the H2O is removed. Hence we can also say the elimination of the water from 2-phosphoglycerate produce the phosphate ester of the enol form of the pyruvate phosphoenol pyruvate PEP, and this reaction also raises the second phosphate residue to a hypotension. The last step of the glycolysis is the transfer of the phosphoryl group from phosphoenol pyruvate to ADP. In this step, phosphoenol pyruvate second is converted into pyruvate second with the help of the enzyme pyruvate kinase. It is the second ATP forming reaction where subtract level phosphorylation also takes place, and here 2-ADP is converted into 2-ATP. In this last step, the pyruvate kinase transfer this residue to ADP, and the remaining enol pyruvate is immediately rearranged into pyruvate, which is much more stable. In this slide, we see all the steps of the glycolysis from glucose to last pyruvate. Here we can also see all types of the enzyme which involve in a glycolysis. The significance of the glycolysis. First point, in glycolysis 1 mole of the glucose is converted to 2 mole of the pyruvate and 2 mole of the NADH by cystosylic enzyme. cystosolic enzyme. Second point, the glycolytic pathway is employed by all tissue for the breakdown of the glucose to provide energy in the form of the ATP. Third point, important pathway of the production of the energy, especially under the anaerobic condition. Fourth point, it is crucial for generation of the energy in cell without mitochondria. And fifth point, it forms products that are intermediate for other metabolic pathway. So it is all about the process of the glycolysis. Thank you.